Welcome everyone to this EACR webinar. I am Alexandra Boitor, Scientific Officer at the EACR, and I'll be your host and technical assistant alongside my colleague Andrew here for this webinar. This is part of our efforts to keep research curious. As you may have noticed already, at the EACR, we're campaigning to keep research curious because we truly believe that early-stage, curiosity-driven cancer research is the cornerstone of future medical breakthroughs. It takes a long time for these foundational discoveries to translate into clinical treatments, but this journey is crucial. To continue to save the lives of future cancer patients, we must continue exploring the unexpected and unknown. Support for this type of curiosity-driven research opens the door to treatment, prevention, and cures, and we can't even imagine yet. This potential for exploration and discovery is what makes our work so vital and promising. So today we invited Dr. Helen Rippon, Chief Executive of Worldwide Cancer Research, Professor Alberto Bardelli, Scientific Director of IFOM, Dr. Sophie Postel-Vinay, physician scientist at Gustave Roussy Cancer Campus, and Dr. Matheus Dias, senior scientist at the Netherlands Cancer Institute. In order to tell us more about their journey of scientific discoveries from bench to bedside. I can't think of any better suited guests to advocate to keep research curious from a range of perspectives from the charity sector to research and clinical practice, bringing together the perspective of early career and senior figures in the cancer research field. And with that, I would like to welcome all of our guests on this webinar. And yes, welcome everyone.
Thanks,
Thank
Alexandra.
you. Thanks for inviting us.
Thank you, a pleasure.
Today we set to discuss the importance of curiosity-driven research, also known as blue sky research. I think it would be nice to begin our conversation with a few examples of how basic research transformed cancer care. My favorite example, I think it's a classic one. I'm fascinated by the impact that the development of DNA sequencing techniques ended up having on cancer care. The genome from cancer cells is routinely sequenced in many hospitals to aid oncologists in treatment decisions. Ultimately, we owe all this to the development of DNA sequencing techniques by Sanger in 1977 and the development of PCR by Karimulis, which relies on an even earlier discovery from the late 1960s when Thomas Brock, a microbiologist, from Cleveland and his undergraduate student conducted research in Yellowstone National Park. And they were driven by the simple curiosity of finding out what forms of life could live in spring waters at 70 Celsius degrees. And that's how they discovered Thermus aquaticus, the organism that had the DNA polymerase and made PCR possible and made the life of a researcher a lot easier in the way. So I'd like to welcome you to share your examples or some more examples of how basic research transformed cancer care, in your opinion. Helen, would you like to go first?
Sure. So, yeah, this is quite a big question, isn't it? But I think what immediately sprung to mind for me were those sort of enabling discoveries that have directly allowed new cancer treatments to be developed. So thinking back to things like the Philadelphia chromosome, which then becomes targetable with Gleevec, to the discoveries around immune checkpoints, which then, brings us the immune checkpoint inhibitors, which really have transformed cancer treatment. And a more sort of personal example, I guess, from worldwide cancer research, because we were involved in some of the very early basic research back in the 90s, was the pulling apart of DNA repair and the concept of synthetic lethality and combining a genetic defect with a drug. And that led to PARP inhibitors, initially olaparib, but there are now next generation drugs coming online. And I think, so when I think about that, and what is inspiring. It's the fact that scientists didn't just have the curiosity to pull apart what is going wrong in cancer cells and how is, for example, cancer evading the immune system. It's being alert to what those serendipitous findings almost tell you about a practical application. So how do we make this a druggable target, even if the technology to create the drug doesn't exist yet? It's that whole line of research which requires curiosity and drive, actually, to keep taking it forward. Otherwise, these things would just they would just remain as interesting findings. So that's where my mind went.
It's interesting to see, and definitely very interesting and important contribution. Sophie, I would be curious to see what's the perspective of a clinician on this.
Well, actually, I fully agree with what Helen just said. And one of the discoveries that came to my mind was also actually the one of PARP inhibitors and synthetic lethality, because I was very fortunate to be at the time of the phase 1 trial in the Johan de Bono's group, where we saw the first responses in patients. And then I joined the ICR group to do my PhD working on high throughput screen synthetic lethality. And I know at the time, lots of people were telling, well, these screens is like a fishing experiment. You don't know what you get out of them, you know, but still there are lots of things which came out of this, which have had clinical implications. So there are the PARP, but also the ATR with ATM and now other compounds, which are really, really coming forward and have changed the lives of patients in clinic. So to me, this is really something in which research played a key role. And where we wouldn't have identified these biomarkers if research hadn't been here, basically, and also hadn't been pushed forward in the clinic by people who believed in preclinical research. Because at that time, phase 1 were very unselected. We put all kinds of patients, and people were like, well, selecting patients, you know, it looked really weird. It was just a toxicity finding thing. And really selecting patients based on preclinical good research data has really changed, I think, the life of patients and the mind of many clinicians as well. So, yeah.
Yeah, that's a good point, Sophie. And now it's very interesting because now, a few years later, screens are actually used pretty much everywhere for this initial discovery of new targets and so on. And these targets are really informing cancer treatments throughout the world. I can even potentially share one first story, just a personal story of how very basic findings could actually be close to translate to the clinic. Because when I was in the end of my PhD, beginning of my first postdoc, I was working actually not even with cancer, but with regulation of the cell cycle. And there was some strange finding in the lab that the lab was using a growth factor, which was actually FGF2. We were using the growth factor to drive the cell cycle, but it turned out that one cancer cell model that we were using, instead of growing more, the cells were growing less. And no one could really understand what was going on. And instead of just discarding this finding, we actually kept investigating to figure out what was actually going on. And then by that time, we found that what was happening is that the growth factor was doing precisely what it was supposed to do, to activate oncogenic signaling pathway. But because it was a cancer cell, this activation was way too strong. So the cells were getting very much stressed out because of this hyperactivation of oncogenic signaling. And then many, many years later, we would fortunate enough to replicate this finding in many different models. And now we are close to set up our first clinical trial to test this idea that you can eventually overactivate oncogenic signaling in cancer cells and combine this with the targeting of stress responses to actually promote an alternative treatment for cancer patients that are essentially out of options for the current therapies. And it really came from a very basic, unexplained finding in the lab. And hopefully in the next coming months, it will be on the clinical practice, at least for a clinical trial.
That's a great example, like in the going. I'm wondering what inspired you in the original research.
The funny thing is that because I didn't work with cancer at the time, so I didn't have this mindset that, oh, it's not possible that the growth factor is actually blocking the cell cycle. There must be something wrong. For me, the data was the data. I was just looking at a cell cycle blockage. Then I was trying to understand what was going on. But then when I actually did, it immediately came to my mind that because all cancer cells would share this defect of having an increased oncogenic signaling given due to mutations or other oncogenic alterations that could be used in many different cancer cell types as long as you find exactly what is the right activator to use in these different cells. And this concept of making a bad situation worse is actually not even new for cancer research. Alberto is actually a specialist on that scenario as well, of making a bad situation worse for cancer cells.
Great. And if our audience is interested in finding out more about Matheus's research, they can listen to our latest podcast episode, The Cancer Researcher. And moving on with this topic, Alberto, I was wondering if you have an example of bench to bedside research that you would like to share with us, or of a like curiosity-driven research that ended up having a strong impact in clinical practice.
Yeah, the main thing, first of all, it's great to be here. And it's great to talk about blue sky. So let me tell you why. I now direct an institute, as you mentioned earlier. And we are all pushed by our founders. In our case, the founder is the Italian Association for Cancer Research, which is the largest European funding body for cancer research in Italy. And we are pushed to go to the patients, which is totally understandable. And that's what I push my PIs. We have 27 PIs here. I push them to do every day and the postdoc. At the same time, we should never forget, and please listen to what we are saying to you, that major discoveries are made sometimes by curiosity, as was alluded to, sometimes by mistakes, and sometimes by looking at something that you don't want to look initially, and then you discover. And so despite what everybody tells you to make a discovery so that you can cure patients, I would tell you today, go and look for whatever you're excited about. And if it is important, it will turn out to be important for the life tree in this planet. And so someone will benefit from it, as we always have been doing on planet Earth. And so, you know, looking at bacteria is equally important, I would say, than looking at patients from some perspective. Now, having provoked the discussion, I can tell you a little bit of what my students now call vintage biology, which is what I was doing when I was a PhD student myself. So they literally created this term, and I told them they wouldn't graduate if they keep using it, but they still use it. So Professor Bardelli performed vintage biology research, but that's okay. And I was at UCL many years ago. And what I studied back then was how to make proteins in Spodoptera frugiperda, okay, SF9 cells. That was my PhD. And you would say, oh, how boring. Yet it was important back then because it was the first time we were able to express receptor tyrosine kinases in an eukaryotic cell. And so we devised new antibodies, antiphosphatyrosine antibodies, and new ways to study cell signaling. So sometimes, you know, you can really put together all parts of the life of tree, on the tree of life, and then learn something. If you wanted to, one more example, very more related to this is, when together with René Bernards, we started to study EGFR signaling in more details. We now know that KRAS is a dominant oncogene that prevents sensitivity to targeted therapies, many targeted therapies, right? I mean, it's a major issue. But we thank those that discovered the signaling pathway, because when we started, at least we knew from C. elegans, from Drosophila, you know, that the pathway was linear, beautiful linear pathway. You know, it's the same pathway, despite some politician thinking differently, that is conserved from, you know, millions of years. And it's just beautiful how it is conserved. And so now, now when we published that at the beginning, people told me it's in Nature, but it's obvious. And I told them, no, it's not obvious, because until we prove it, it's not like that. But interestingly enough, a discovery that was made possible thanks to the work done in nematodes, in yeast, and sometimes in insect cells, turned out to be very relevant for understanding signaling in cancer cells that is now at the root of developing new therapies for melanoma, for example, for colon cancer, for lung cancer. Without that, we wouldn't have a chart of the signaling pathway, and we wouldn't be where we are.
Thank you, Alberto. And to continue on that note, if anyone from our audience is interested in finding out more about the research that Alberto cited, that was performed in his lab together with René Bernard's lab, we have a webinar as part of this same campaign to keep research curious on YouTube with René that touches on that subject as well. But continuing on this topic, Alberto, you mentioned something that made another question pop into my mind. So would you agree that the past few years have seen an increasing global trend among funding agencies, and not only, to shift their budgets towards translation and clinical discoveries? And don't get me wrong, as you said, I don't by any means want to dispute or undermine the importance of translational and clinical research, but I'm wondering if there's a concern that funds are shifted away from basic research instead of being supplemented. And I would like to get your views and the views of all panelists on this as well.
Thank you. I mean, I think it is a real concern. And it's driven by goodwill, which is to get back to patients as fast as possible. At the same time, nothing drives discovery as curiosity. And if you ask your postdoc, your PhD to study a biomarker, I mean, perhaps their mindset will be slightly limited by going forward in identifying a specific, a specific biomarker. At the same time, what I recommend to those that are listening is that this doesn't change anything, because even in a very pragmatic, clinically oriented funding strategy, you still have opportunity to develop curiosity-driven research. A few days ago, we were looking at Western blots, and at some point there was a shift in a band, and that shift was generated by using a new drug. And the student said, you know, I need to repeat the Western blot because there is a shift. And the reality is we discover how to measure exactly the engagement of the new drug with the human tissues. So it was super exciting, and mouse tissue. So, you know, what I would recommend is it is a real concern, and ERC and other funding agencies are trying to supplement that. Thank God we have ERC in Europe, right? This ERC opportunity that is great and allows you to, you know, really develop excellent science without necessarily being driven by outcome, like clinical outcome or support to patients, exist. But at the same time, you still have to opportunity to do what I often call secret projects. So I recommend that anybody that is listening starts immediately a secret project on curiosity-driven research on what he's doing or she's doing. Don't tell your PIs until you have found a major discovery, and then go back to them. Anyway, hope this stimulates the discussion.
Yes, and good advice for early career researchers. Helen, I'm interested in hearing your perspective and worldwide cancer research perspective on this. Is that something that you are also concerned with, or something that you've noticed?
Yeah, definitely. So we exist, and we were set up by a scientist 40 odd years ago to support blue sky curious research. We are here to seed fund new ideas and get new stuff off the ground, not really to fund existing lines of research, because there's plenty of funders who can do that. We are concerned about the trend of funders turning to more translational clinical work at the expense of discovery science. I can understand why, right? So it's hard to raise money. We're a charity. It's really, really hard to raise money, and it's easier if you can show direct short-term impact. that said, I think we find at Worldwide Cancer Research, like, people understand if you explain the fundamentals, that you don't end up with any new thing in the world, you don't end up with a new cancer treatment, you don't end up with a smartphone unless you've got the basic technology and the science that underpins it. And somebody has to back that initial idea. People do understand it. So I guess what I would say, as coming from a funder of, we will fund research into any cancer. In fact, it can just be basic cell biology that might tell us something about cancer. We will fund anyone, anywhere in the world, so long as you're actually a scientist, the only requirement that we have is its innovation and discovery, and it could tell us something fundamentally new, and we get that a lot of those projects will not turn out the way that it was initially expected, because that's research. What I would advise people on this webinar to think about is asking a really, really good question. That's what we're interested in as a funder, is like, ask a really important question. So a project that we recently funded in the last grant round was actually a project in Italy, Serena Zacchigna. I hope I've pronounced that correctly. And she's just asking, why does the heart not get cancer? It's such a basic question. And what we often hear with our scientific committee when they fund these projects is they read them and they go, I wish I had thought of that. It's a great question. And take your research that way. Ask something important. And I wish I'd known that. So I started out as a scientist, and I, fell out of science after about 6 years of postdocing. And I wish I had known then what I know now, that it's about the question.
That's beautiful advice. Matheus, I can see that you raised your hand. Do you want to continue on Alberto's note? Tell us if early career researchers have enough time for like side projects.
No, also that, but just to complement what Helen is just saying, it's very important to have this kind of funding, because we can imagine that if you have a very translational project that is translational from day one, it's probably built on some knowledge that is already there. And most of the knowledge that is already there, if they would deliver real, clear, transformative benefits for the patients, they would have done it already. So maybe it's important to ask indeed good questions, different questions, because then we can potentially later translate these different questions into really different treatments. Otherwise, we would be just trying to make incremental improvements in cancer treatment, but we might not really get where we want to be as a cancer research scientific community.
Thank you, Matheus. Sophie, what's the perspective from your perspective?
Yeah, I completely agree, and I have noticed this kind of drift again as well. I mean, regarding from very basic open blue sky research to more applied or translational research, which is obviously really good for patients. But I think, I mean, to me, they have completely different aims and also time scales. And sometimes we see grant refused just because rejected, just because they say, well, this will take ages to come to patients, meaning more than 10 years. But I think this is what good research takes as well. You have to be realistic. And you cannot discard a good project just because you're not sure of how long it's going to be to take to translate to patients. And also lots of discoveries that have been having impact in patients' lives have been made by serendipity. So now we are asking to write grants where we put expected outcome and expected results at the end of the 5 years. I'll get this done. And I think this is somehow restricting your mind, because you feel forced or obliged to deliver these bullet points on top of your secret project, which I think many people do have. And so I'm not saying it's a bad thing, but I think it's good that we have different grants with different aims, and you have to really pinpoint the right scheme according to what's your type of research doing. And with regards to the curiosity as well, I think it's really important that younger people do share every result with the PI, at least they want to share, because sometimes they do not get the expected results. And they think, well, oh, actually my experiment is wrong. But no, if the controls are correct, the results should be correct, and you should trust your results. And maybe you have discovered something really interesting, unexpected. So I think it's really important to be open-minded, discuss, and also I know it's EACR, but potentially discuss with people beyond cancer, beyond your specialty. We see we have learned a lot from immunologists, from geneticists, from metabolic people, from also human disease like interferonopathies. They were very insightful into the interplay between DNA damage repair and cancer and the anti-tumor immune response. So I would encourage people to keep their mind open, discuss with people from other specialties. And I think, to me, basic research, translational research, and clinical research all really complement each other. And it's good to have grants dedicated to different types of research.
Thanks, Sophie. You made two really good points about the grant review process and the need for collaboration across research fields, which links straight into my next question pretty much. How do you think strategic paths should be established for cancer research?
I think, again, it should be between a collaboration from basic researcher, translational clinicians, and as well patients, and of course funders, because everyone has different expectations. And this can be really broad, going from very basic questions to quality of life questions, which can also benefit from basic research sometimes. And so I think this should really be, as Helen rightly said, you need to have a relevant or good question, which drives everyone, and then try to get as much, feedback you can from different people thinking differently. And this can give rise to very original projects, I think, which can make an impact.
Alberto, would you like to try and tackle this question next?
Yeah, I mean, I'm not sure I can add much. It'd be interesting to know what those that are listening to us, maybe they have stories to share and other aspects, but there is not different type of research. As Helen put it very beautifully, the most important thing is to ask relevant questions. If you pick relevant questions, as we always say, whatever you do, your project will be absolutely stellar and maybe challenging, but in the end will be very relevant. If you settle down for a non-critical question, then you may end up with much less impactful research. And that applies to basic translational clinical. I don't think it makes a lot of sense to separate these type of things. But overall, it's true that in the last 10 years, I've seen more and more funding bodies pushing us towards translational research. And I do this in my institute because that's what we are, pushed to do. But we need to protect these bright minds. And after all, I don't think that when immunotherapy was discovered, someone was thinking to cure someone. Someone was trying to understand how the immune system was working in mice. And we have to thank these people that kept doing so. Yeah.
With that, Helen, is there anything else you would like to add in addition to highlighting the importance of a good question?
I think on this issue of strategic priorities, I really grapple with this as a funder, because I worry that when you start setting strategic priorities, number one, that's inherently backward looking, right? That is in some ways, well, no, in all ways, based on the things that we already know. And as a funder of curiosity-driven blue sky research, we're really looking ahead at what don't we know? What are the future lines of research that are going to lead to treatments that we can't even conceive right now? So I have a bit of a worry about strategic priorities from that perspective. And then the other thing I worry about, and it's one, and we do sit as a bit of an outlier amongst funders in this respect. But when you start setting strategic priorities as a funder, you then create gaps, right, between things, because you scientists have to crowbar your research within the parameters that have been set by funders, and then there's gaps, right? Inherently, there will be gaps between. So worldwide cancer research, we don't set priorities, we don't define, we want to see things from particular disciplines. In fact, we love to see discipline hoppers who are coming from things other than cancer research, like developmental biology, for example. I think there's a theme emerging here about question, isn't there? So there might be something around, like, innovation often comes out of just getting people together to have a fairly unstructured chat about what's important and to start thinking broadly and laying options on the table, even things that seem completely crazy and undoable. Well, let's just stick it all out there and have a bit of fun. And I think funders often refer to them as sandpits. So that kind of approach, I suspect, can work when you sandpit an idea, but you need to get, all the right people in the room with lots of different perspectives coming from lots of different disciplines. That's something I suppose I'd like to see more of. But that's not about going as a funder, well, we want to fund new immunotherapy research to the exclusion of other things. So does that make sense? But we are a bit unusual, I think, as a funder in that way.
Does make sense, and I'm sure that everyone appreciates every now and again for someone to be a bit different and address like that group that maybe others haven't thought of. So I'm sure your approach is very welcome.
I should probably say at this point, we have a grant round open right now. It closes on the 31st of March. So if anyone's got something in their head that they don't think they can get funded by anyone else, it's that, like, that question you've always wanted to ask, that set of experiments you've always wanted to do, then do take a look on our website, because we have a round open right now.
Sounds like a great opportunity. So if anyone is interested,
if we had a few more days on the deadline, I would definitely apply after all this amazing talk about crazy ideas.
I'm sure you can rattle off an 8-page project proposal in 3 hours in your office.
Okay, sounds like a plan. I might.
Sounds like a challenge.
I'm gonna have a few hours on the plane tomorrow, so why not? No, but it's a very important thing what Helen is saying, that I think we as researchers, we need to keep our minds open, like to different ideas, like to different, and just like Alberto just said, and also Sophie, it's important to let the data guide our research, even though it might be an experiment that we don't know exactly what to expect, or that the result came out is actually very different from what we expected. But we can really turn this into something beautiful and something important and something interesting, but just by following the data with our minds fully open. It's not because we have a hypothesis that the data or the biology itself should conform to our boxes and hypotheses. Let's keep our minds open and just follow the data.
As Alberto mentioned earlier, we would love for this webinar to be interactive. So we invite all of the participants, the attendees, to type their questions in the Q&A box. And something I might have forgotten to mention at the beginning, you will have the option to ask your question out loud, join our discussion panel for a few minutes and discuss your questions. So please do use the Q&A box to share your thoughts with us. And I'll take this opportunity to thank the participant that is thanking us for the webinar. We are happy that we managed to make you feel a bit more confident about your research. And whilst our attendees are trying to think of a few questions of their own, I was wondering if you could give me a few examples of what the curiosity plays in your work currently. So the role that the curiosity plays in your work currently, and how do you make space for curiosity-driven research in your organizations? Matheus very nicely started answering this question earlier, I think during the first question of the webinar, really. But I would like if everyone else could chip in as well.
I can
Sure, Alberto.
I can try if you wish. So one thing we did inside our institute this year is to start what we call Archimedes. So Archimedes are very small grants that we give internally with a very tight competition, and the PIs cannot apply. Only students, technicians, postdocs can apply. If they get the grant, they pay back their salary to their PI, and they are free to do what they want for 6 months. So this has been a little disruptive, as you can imagine, but it's brought forward 11 applications. This is we are in institute around 350 altogether. And some of them are really, really unexpected, okay? And so one way to turn this around is to see whether the youngest might have bright ideas and questions they want to address, and offer them the opportunity to work a little bit independently at the very beginning on their own project. So that's one thing we taught. And our community is just completed, and the 3 winners move forward. What I mean, I guess, is allowing very young people to have the opportunity to develop their own ideas is very important from my perspective.
Thanks, Alberto. Sophie?
Yeah, I fully agree with this, and I think that it's important to have programs as well for young investigators to really develop what they want and give them more freedom. With regards to maintaining curiosity, well, I think, as I said, it's really also exchanging about unexpected results. You can learn a lot from an unexpected result. Keeping reading, going to Congress and meeting people, not being afraid of talking to people, because this is often when you learn the most, because some people have been working for years on something and they can tell you, well, this is super interesting because of blah, 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 and you would have completely ignored it. And this is also true for a clinic. For example, when you see an unexpected response to a drug in a patient, it tells you something. It's not that you may not have the right molecular biomarker, like sorafenib is one example. It was developed as a RAF inhibitor, and it worked really well as an anti-angiogenic in renal cell carcinoma. And this is just curiosity, noticing interesting observation, discussing them out of your specialty. And I think, yeah, this is a nice way to encourage people to maintain their curiosity. And also sometimes making some smaller groups for people to present their project in a very few minutes, but just in a nutshell, and favoring exchanges. I think sometimes this favors interesting and original ideas and collaboration between groups.
Thanks, Sophie. Helen, you mentioned already that Worldwide Cancer Research is offering perhaps a bit more funding than other organizations to Blue Sky Research. Is there anything else you would like to highlight that your organization does to support curiosity-driven research? Or perhaps tell us how curiosity helps you in your line of work?
Well, I can do both. So it's probably worth saying that we partner with the EACR to offer travel fellowships to young researchers. So to help them go to other labs, go to conferences, and just, have those conversations and learn from people with a different set of experience and perspectives. In Worldwide Cancer Research, curiosity is actually our lead cultural value, because we want to embed the curiosity in our scientific program of funding right across the organization and across everything we do. And I find that there are 2 probably key things there. One is creating an environment where people can talk about their ideas and talk about their mistakes and the things that have gone wrong in a non-judgmental way, and in a supportive way. And I think it's, as Sophie and Alberto, I think, have already alluded to, if you don't have that, then particularly as a young person in early career, you might hide the things that you feel aren't right. So I think part of a really important part of stimulating curiosity is creating the right environment for people to be open and honest. I also think there's an element of creativity loves a bit of constraint, right? Or a bit of urgency. So there's a reason that technological progress is greatest during wartime, right? And I'm not suggesting we go to war, but actually presenting some challenge around, you know, so if I were to think about often our biggest challenge as a charity is fundraising, it's generating the income. So it would be thinking about, okay, well, we have this much to invest in a fundraising campaign, or this much to invest in a new piece of tech that, you know, might help us reach more supporters, or, you know, like, how would we use that money? And sometimes it's presenting a little bit of constraint, which spurs people to be innovative, I think. So those are the two things I would throw into the pot here, which may or may not be relevant to scientific research in a lab.
Thanks, Helen. I can see that our conversation prompted a few thoughts from our audience. I'll read out loud the questions that we received because they are all anonymous. So someone's wondering, do you think academic science as it functions today is well set up for curiosity-driven science? Are there any changes you would like to see in the industry? Anyone dare to approach this question?
It's difficult to comment, but one suggestion I would have is, if you are asking a great question, I think you'll attract attention, and I think it may be possible for you to start your own and develop it further. I mean, sometimes great idea will spin off. And so one way to address the question is to change the culture. Maybe you can be the first to believe that you can, you know, turn it around and come up with something brilliant. So don't be ashamed, or sorry, don't be afraid to initiate at some point of your career your own idea throughout the development, like a biotech or something like that.
Thanks, Alberto. Someone is asking if anyone could help share like on how to start on initiative of novel research initiative on how to manage to implement in limited resources settings. So I think this is somewhat similar to what you were saying earlier, Alberto, that you had like a new initiative in your institute to try and promote curiosity-driven research. This person, I think, would like to do something similar, create like a new initiative, because in the same message they say that they feel that their country is not supporting research enough. It would probably be easier if we knew what the country in question was.
Yeah, like you said, it's a little bit difficult to address the question because I'm not sure I fully get the context. But maybe this person can certainly find, if they want to talk to me, they can find my address and maybe send me a private message, and I'd be happy to address this further. Yeah,
so either that, or please just like, if you could give us a few more details in the Q&A box as another question, that would be really helpful. Thank you. Another person would like to ask the panel if you feel that blue sky research is particularly of relevance to rare cancers. Generally, rare cancers are so under-researched. What we find in them often goes completely against the rules that are based on common cancers. This person goes on to highlighting. Sophie, I can see you're nodding. Do you want to address this question?
I agree with that, and also wanted to potentially comment on the previous question with industry, and I think it goes together. I think industry and academic might have different, aims, and you have a liberty and freedom of thinking in academia that you might not have in industry because they have strategic priorities. These strategic priorities can change quite rapidly. And this can kill some research projects quite rapidly as well, which can be very disappointing for some scientists. And also industry will not study rare cancers or because just there is no market or limited market. And still, these diseases are extremely interesting because they are really good models. Sometimes they are created by one unique alteration, which can drive cancer. No one knows why. And you can really learn a lot. So using them as a model, to me, is really the role where academia can also bring something. And academia can also collaborate with industry. Many industrial partners now have foundations where they offer to fund independently of their own research, academic programs in collaboration. So I think we should not oppose both. They may, have different settings and different strengths and different weaknesses, but I think it's a good opportunity to collaborate. And definitely rare cancers should be studied because you can learn a lot from them. And yeah, even I agree, it's more difficult to work on rare cancer than common cancers for many reasons. But I would definitely encourage the person who asked that question, if she or he is interested, to carry on his or her work.
And it's pretty clear that the person knows a lot about rare cancers, because one thing that it's very common in rare cancers is that they have a very low mutation burden, especially those ones from childhood. Sometimes people don't even find any oncogenic alterations that could explain the phenotype. So it's clear that we can learn a lot from those rare cancers, because perhaps all the mutations that we are seeing in other cancers as well, they have a role that it's not as big as we think it is. So we can have many different alterations leading to the kind of phenotypes that we are observing in the more common cancers. And we can learn a lot about this from studying these rare cancers.
Thanks both of you for your answers. The next couple of questions, I think they would go together quite easily. And they are more like career related. The question is if you think that there's a place in the European research for PhD students who work on fundamental research and what they could expect from their career. With the next participant going on into like detailing, I think this question or like a similar question a bit more, asking if a PhD student should try challenging curiosity-driven research, if there is a 3-4 year time limit and stipend limit to publish at least 2 first author articles before they are allowed to defend their PhD thesis, or if it would just be better to pick up a simpler and straightforward project in order to try and get their PhD finished in time rather than focus on the curiosity-driven research. So the way I see it is pretty much like how would you balance as an early career researcher or a PhD student the research question that you would like to follow because you are curious about with the time constraints that are imposed by your institution. That's how I read these two questions, so that it wouldn't affect your career progression too much. Anyone that would like to answer? Perhaps someone that deals with PhD students on a regular basis. Alberto, Sophie, like you are supervisors. What do you say to your own students?
I think I said most of the things that I conceive about this topic. But when a student joins my lab, the 3 keywords that he gets or she gets are pick a relevant problem, focus, and think papers. This is how they are sort of instructed to follow. And the first one is the same that Helen pointed out at the beginning. So I also tend to be very, very very unfocused research sometimes, because that's how discoveries really are made. I believe in this. I mean, I've seen people in the group that and students that were able to develop projects that were not initially what they thought, and because they had initiative. So, but other than that, it is very much also to the students to be able to do that.
Yeah, I fully agree. And I think the drive is very important here, which is why we should kind of be guiding the student, but not be too restrictive either, because if a student becomes passionate about something and the idea is great, there is no reason why, at least from what I think, I would refrain him from pushing that as much as this. I feel this is reasonable. And the PhD students that have, my experience is not illimited, but from the few I could supervise, the ones that did the most were really the ones that went into their project, they took the time to read, to make it their own. And as we said at the beginning, remain curious, unexpected things can happen, but had this drive of not for a publication, but for science, you know, I mean, of course, publication is what you need at the end, but also just because they like what they do, they are driven and they will succeed. I think, yeah.
Thanks, Sophie and Alberto. I'll invite now Margherita Perillo to ask her question. Margherita, are you available?
Yeah, can you hear me?
Yes.
Thank you. So yeah, thank you everybody for this panel. It was very informative. My question is, so we are talking about basic cancer research, and I'm a developmental biologist, and lately by doing the curious science, I ended up discovering some mechanisms that can be also important in cancer, specifically in EMT and invasiveness. So I was wondering, like, what do you think about using studying basic research, but not using like cell culture or mice, but using alternative non-mammalian systems that share a lot of basic mechanisms with, mammals, but are not used nowadays for cancer? Like, do you think there are grant funding sources that are also interested in, bringing to the table like new systems, or do you think it will be risky to work also, on non-conventional systems?
Margarita, thank you first of all for bringing this forward. It's an interesting topic, but I'm not sure what you propose. So are you suggesting like Drosophila yeast, C. elegans, or something completely unrelated to that? And if so, what do you have in mind?
So, well, I actually work with C-star embryos and larvae. So it's not flies, not nematodes, but these are animals that are really share the same mechanism in embryogenesis and organogenesis like vertebrates. And I just wonder, if it's something so new, and I see some mechanisms or EMT, and I discover a new protein, for instance, right? Do you think that it's something that one can think of applying also for cancer research?
I would encourage to try. I mean, I think it's in principle a great idea, and fundamental mechanisms are shared as we discussed at the very beginning. So I think you'll make important discoveries as long as you have a model that you can perturbate and you can study properly. I think I encourage you to do so. Yeah.
Thank you.
I really agree. And I think one example, coming back to the first response, was the synthetic lethality. Many of them, and the most potent one, can be shared across many species and even yeast and so on. And of course, you cannot extrapolate everything to humans, but I don't think this should be a blocking step, at least.
So I would add that would be within remit for us as well, potentially. So we have definitely funded things on sort of fundamental cell biology that were not in mammalian or even vertebrate systems. So yes, I would also encourage you to pursue it, because it sounds like you could bring something really new with a new model.
Well, thanks everyone for such a lovely conversation. I think we uncovered a few very inspiring stories and great examples of building up on basic research discoveries to groundbreaking novel drugs. I apologize to everyone that typed the question and we didn't manage to get to answer it. But we are approaching the end of the webinar. We want to take into account your time and our panelists' time, so we need to bring this webinar to an end. Stories like those that were shared today make us at the EACR feel excited and optimistic about the future of cancer research. In order to celebrate the incredible potential of basic research, we introduced a new plenary session at the EACR Congress, From Bench to Bedside and Back. If you're interested in the latest developments in cancer research, join us for the EACR Congress on 16-19 June in Lisbon. Early rate registration deadline is in a month on 28 April 2025, so that would be the deadline. Check the program at 2025.eacr.org to learn more about the topics that will be addressed during the EACR Congress. If you liked this webinar, you might also be interested in episode 8 of our podcast, Freeing Scientists to Explore the Role of Curiosity in Cancer Research, with Professor Ruth Palmer from Sweden, Professor Daniel Pepper from the Netherlands, and Professor Iwona Lugowska from Poland, or the blog articles in our magazine, The Cancer Researchers, highlighting the critical role of curiosity-driven cancer research. You can find a link in the chat to all these Keep Research Curious related articles. Let's keep the spirit of curiosity alive and thriving. Helen, Alberto, Sophie, Matheus, is there anything else that you would like to add?
Just thank you for a really stimulating discussion.
Yeah, thanks to all participants for their input as well.
Yeah, it was amazing. And as we can see on the questions here, we already made at least one PhD student to promise that he or she will be forever curious, which is already a win. It's already a win.
Yeah, I agree. So thanks again to our panelists, and many thanks to everyone who attended. I hope to meet some of you at the Congress in June. Have a nice day. Bye-bye.
Bye. Bye-bye. Thank you.
Bye-bye.